Good morning, everyone, and thank you for being here today. I'm excited to introduce our presentation on ESKAPE pathogens. My name is Dr. Mayuri Rani. Today, we'll be exploring these critical pathogens, their impact on healthcare, and what we can do to combat them. We'll cover their characteristics, resistance mechanisms, and strategies for prevention and control. Let's get started. So, what are ESKAPE pathogens? Well, this term refers to a group of bacteria that are particularly notorious in healthcare settings. These microbes are known for their ability to escape the effects of many antibiotics, making infections they cause especially challenging to treat. The name ESKAPE actually comes from the first letter of each pathogen. Enterococcus faecium, Staphylococcus aureus, Klebsiella pneumoniae, Acinetobacter baumannii, Pseudomonas aeruginosa, and Enterobacter species. These pathogens are significant because they are often responsible for severe hospital-acquired infections like bloodstream infections, pneumonia, and wound infections. They tend to be highly resistant to multiple drugs, which is a major concern for patient outcomes and infection control. Understanding these pathogens is crucial because they exemplify the growing threat of antibiotic resistance, something that ties directly into our overall focus today. Recognizing the key players in resistant infections helps us develop better strategies to manage and prevent these tough infections in clinical settings. In February 2017, the World Health Organization took an important step by publishing a list of pathogens that urgently need new antibiotics. This list was designed to help focus research and development efforts on the most critical threats to global health. Among these, there's a group called the ESKAPE pathogens, which stands for Enterococcus faecium, Staphylococcus aureus, Klebsiella pneumoniae, Acinetobacter baumannii, Pseudomonas aeruginosa, and Enterobacter species. These bacteria are particularly concerning because they tend to be resistant to multiple antibiotics, making infections difficult to treat. By designating these as priority, the WHO highlights the urgent need for new antimicrobial agents to combat these dangerous pathogens. This effort ties directly into our overall theme of addressing antimicrobial resistance and the critical need for innovation in medicine. Let's take a closer look at how ESKAPE pathogens develop resistance. They do this primarily through genetic mutation and the acquisition of mobile genetic elements, or MGEs. These mechanisms enable them to adapt quickly and evade our antibiotics. You'll notice that these bacteria have become resistant to many classes of antibiotics, including oxazolidinones, lipopeptides, and macrolides. They've also developed resistance to fluoroquinolones and tetracyclines, which are commonly used antibiotics. More concerning is their ability to resist lactams and lactamase inhibitor combinations. These are often frontline treatments, and resistance here can severely limit our options. Even resistance to last-line antibiotics, such as carbapenums, glycopeptides, and polymyxins, has been documented. These drugs are typically reserved for the most resistant infections, so the development of resistance is quite alarming. However, it's important to note that resistance to lipoglycopeptides remains rare and has only recently been observed. This suggests that these antibiotics are still largely effective, but we need to stay vigilant. Overall, understanding these resistance mechanisms is crucial as we continue to combat antibiotic resistance and develop new strategies to stay ahead of these adaptable pathogens. Today, we're focusing on the mechanisms of antibiotic resistance, specifically in the ESKAPE pathogens. These bacteria are notorious for causing difficult-to-treat infections, largely because of their ability to evade antibiotics. So, what are some of the key ways they do this? First, they can produce enzymes that inactivate antibiotics, like beta-lactamases, which break down penicillins and cephalosporins. This is a common resistance strategy, essentially neutralizing the drug before it can do its job. Second, they can alter their target sites. By changing the structure of the molecules that antibiotics bind to, these bacteria prevent the drugs from attaching effectively, rendering them ineffective. Third, some pathogens increase their efflux pump activity. Think of these as tiny molecular pumps that actively expel the antibiotic out of the bacterial cell, lowering the drug's concentration inside and allowing the bacteria to survive. Fourth, they can develop or acquire resistance through genetic mutations or horizontal gene transfer, rapidly spreading resistant traits across populations. All of these mechanisms highlight the adaptive prowess of ESKAPE pathogens and underscore why combating antibiotic resistance remains such a critical challenge in modern medicine. Understanding these mechanisms helps us develop better strategies to counteract their defenses. This slide doesn't contain enough information. That's why we're moving to the next slide. Let's now turn our attention to antibiotic inactivation or alteration. This is one of the main ways bacteria can resist antibiotics. Essentially, bacteria have developed mechanisms to neutralize the drug before it can do its job. For example, some produce enzymes that destroy the antibiotic, rendering it ineffective. A classic case is beta-lactamase enzymes that break down penicillin and related antibiotics. This process is crucial because it directly impacts how effective our treatments are. When bacteria can inactivate the antibiotic, it means higher doses or different drugs might be necessary. It also highlights the importance of understanding these resistance mechanisms so we can develop better strategies to combat infections. In the context of overall antimicrobial resistance, inactivation and alteration are key players, reminding us why ongoing research and prudent antibiotic use are so vital. Let's take a closer look at target site modifications. This concept is all about the specific changes we make at the target site to improve our outcomes. These modifications are crucial because they allow us to tailor interventions precisely where they're needed most. For example, in genetic editing, this could mean altering a particular gene sequence to correct a mutation. In drug delivery, it might involve changing the surface of a nanoparticle to enhance targeting to cancer cells. The key point here is that by focusing modifications at the target site, we can increase effectiveness and reduce unintended effects. This aligns with our overall goal of making treatments more precise and personalized. So, understanding how to effectively modify these target sites is fundamental to advancing our approaches and achieving better results. Beta-lactam antibiotics work by inhibiting bacteria, and they do this by binding to PBP enzymes, which are anchored in the bacterial cell wall. This binding disrupts cell wall synthesis, leading to bacterial death. However, in MRSA, the story is a bit different. Resistance to methicillin and other beta-lactams occurs through the expression of a foreign gene called MEK-A. This gene codes for a protein called PBP2A, which is a modified version of the usual PBP enzymes. What's important here is that PBP2A has a low affinity for beta-lactam antibiotics, meaning these drugs can't effectively bind to it. As a result, most beta-lactam agents become completely ineffective against MRSA. This is a classic example of how bacteria can modify their target enzymes, what we call target enzyme modification, to evade antibiotic action, making treatment more challenging. And this highlights the importance of understanding resistance mechanisms in developing effective therapies. Now, let's talk about a major way bacteria like S. aureus and Enterococcus spp. Resist macrolide, lincosamide, and streptogramin B antibiotics. This resistance is primarily mediated by enzymes called ermine-coated rRNA methyltransferases. These enzymes work by adding methyl groups to a specific spot on the bacterial ribosome, specifically the A2058 residue within the 23S rRNA of the 50S subunit. This methylation impairs the binding of MLSB antibiotics, making them ineffective. It's important to note that the expression of the ERM gene can be either constitutive or inducible. Constitutive expression means the bacteria always produce these methyltransferases, leading to cross-resistance across all MLSB agents. On the other hand, inducible strains only produce the enzyme in response to certain antibiotics, like erythromycin, clarithromycin, or azithromycin. These strains become resistant to these 14 and 15-member macrolides, but still remain susceptible to lincosamides and streptogrammins. Understanding these mechanisms is critical because they help explain why some bacterial infections become resistant and how we might overcome or prevent that resistance. This resistance, especially through target site alteration, is a key concept in antimicrobial resistance and impacts our approach to treatment strategies. One of the most significant resistance mechanisms that has emerged in gram-positive ESKAPE pathogens over recent decades is glycopeptide resistance. In susceptible gram-positive bacteria, glycopeptides normally work by inhibiting bacterial cell wall biosynthesis. They do this by targeting the DLA peptidoglycan precursors that are found on the outer cell wall. But when it comes to resistant enterococci, things get more complex. They acquire specific gene clusters known as Van genes. These genes coordinate two main processes. First, they lead to the synthesis of modified peptidoglycan precursors. These modified precursors have altered termini, replacing the natural DLA with molecules like DLA-D-lactate or DLA-D-serine, which reduces the binding affinity of glycopeptides. Second, the bacteria produce enzymes called decarboxyptidases. These enzymes help by removing residual natural DLA precursors from the cell wall, further decreasing the effectiveness of the antibiotics. Essentially, these modifications in the cell wall precursors are a clever way for bacteria to evade glycopeptide action, making resistance a major challenge in treatment. Let's talk about reduced antibiotic penetration and accumulation. This is a crucial aspect because, in some infections, bacteria develop mechanisms that prevent antibiotics from effectively reaching their targets. When antibiotics can't penetrate the bacterial cell wall or membrane properly, their ability to kill or inhibit bacteria diminishes significantly. Additionally, some bacteria have active efflux pumps that remove antibiotics from their cells, further reducing drug accumulation inside. This means that even if the antibiotic reaches the bacteria, it might not stay long enough or in sufficient concentration to be effective. These factors contribute to treatment failures and can lead to persistent infections. Understanding this concept helps us see why simply administering antibiotics isn't always enough. It's essential to consider how bacteria can block or reduce drug penetration and accumulation. This ties back to the overall theme of antibiotic resistance, highlighting the importance of developing strategies to overcome these barriers. Let's take a closer look at other mechanisms and survival strategies that organisms use. These strategies are often less obvious, but just as crucial for their persistence in challenging environments. For example, some species develop behavioral adaptations like migration or hibernation to escape unfavorable conditions. Others might produce protective structures, such as shells or thick skins, to shield themselves from predators or harsh elements. Additionally, many organisms employ physiological strategies, like producing antifreeze proteins to survive in freezing temperatures. These mechanisms highlight the incredible diversity of survival tactics in nature. They also connect back to our overall theme, how life persists through adaptation and resilience. Understanding these strategies broadens our appreciation for the complexity of survival and the ingenuity of living organisms. Now, let's take a closer look at mobile genetic elements that confer antimicrobial resistance. These elements are fascinating because they play a crucial role in how bacteria quickly adapt and survive in the presence of antibiotics. Essentially, they are pieces of DNA that can move between different bacteria, spreading resistance genes rapidly across populations. These mobile elements include plasmids, transposons, and integrins, each with unique mechanisms for transfer. Plasmids, for example, are circular DNA molecules that can replicate independently and often carry multiple resistance genes, making bacteria resistant to several drugs at once. Transposons, sometimes called jumping genes, can move within and between DNA molecules, facilitating the transfer of resistance traits. Integrins are genetic platforms that can capture and express gene cassettes, including those responsible for resistance. What makes these elements particularly important is their ability to accelerate the spread of antimicrobial resistance across different bacterial species and environments. This mobility not only complicates treatment options, but also highlights the importance of monitoring and controlling these elements to combat resistance effectively. Connecting this to the overall theme, understanding mobile genetic elements helps us grasp how resistance can disseminate so rapidly, emphasizing the need for comprehensive strategies to limit their spread and preserve antibiotic efficacy.